And then finally, transport in the xylem. Endodermal cells and also living cells within this vascular cylinder discharge water and minerals into their walls, apoplast, and the xylem vessels then transport the water and minerals upward into the chute system. And here you can see the pathway along the apoplast and the symplast. Okay. So much of the absorption of water and minerals occurs near the root tips, where the epidermis is permeable, hydrophilic to water, and root hairs are located, increasing surface area absorption. Most plants, extinct plants like early plants that are still living today, conifers, have mutually beneficial relationships with fungi, which facilitate absorption of water and minerals from the soil. So this fungi outside of the roots. And this is mycorrhizae. Okay, roots and fungi form mycorrhizae, symbiotic structures consisting of plant roots united with the fungal hyphae. Okay, so this increases surface area tremendously so that more water can be absorbed. And this was an evolutionary adaptation for these early plants when they grew in very nutrient-poor soil. So plants lose an enormous amount of water through transpiration, loss of water, vapor through the leaves, and other aerial parts of the plant, so the transpired water must be replaced by water transported up from the roots. So xylem sap rises to heights of more than 100 meters in the tallest plants, tallest trees. Is the sap pushed upward from the roots, or is it pulled upward by the leaves? So root pressure. At night, when the stomata are closed and transpiration is very low, root cells continue pumping mineral ions into the xylem of the vascular cylinder, into the steel, lowering the water potential. Water flows from the root cortex, generating root pressure. So root pressure sometimes results, if it's too great, in guttation, like in the early morning when the stomata open, the exudation of water droplets on the tips of grass blades or the leaf margins of some small herbaceous eudicots like the strawberry. So the root pressure is forcing excess water from this strawberry leaf. It's called guttation. So water is pulled upward by negative pressure in the xylem. So here's the transpirational pull. So in transpiration, water vapor, shown as these little blue dots here, diffuses from the moist air spaces of the leaf to the drier air outside the stomata. Okay, so it's moving out here. Moving out. At first, the water vapor lost by transpiration is replaced by evaporation from water film that coats the mesophyll cells. Okay? You can see that there. And the evaporation of the water film causes the air-water interface to retreat farther into the cell wall and becomes more curved right through here. This curvature increases the surface tension and the rate of transpiration. Then the increased surface tension here pulls water from surrounding cells and spaces. Then water from the xylem is pulled up right here into the surrounding cells and air spaces to replace the water that was lost. Okay? So transpiration produces negative pressure or tension in the leaf, which exerts a pulling force on water. Okay, so this is a low rate of transpiration, negative 0.15. This is high rate of transpiration, negative 10. This is the air-water interface, the cell wall, and the airspace here. So it diffuses down its water potential gradient and exits stomata. So cohesion and adhesion of water molecules in the ascent of xylem sap, because xylem is primarily made up of water. Okay, so here is root xylem 0.6, negative 0.6, up here negative 100. So this is greater than that, so the water potential gradient here, it goes from greater to lesser. So transpirational pull is facilitated by cohesion and adhesion. So here's water uptake from the soil. Okay, water uptake. Cohesion by hydrogen bonding of the water molecules and the cell wall. And transpirational pull up through here through the xylem, out through the mesophils and the stomata. Okay? So hydrogen bonding forms an unbroken chain of water molecules extending from leaves to the soil. The force driving the rise or ascent of xylem sap is a gradient of water potential. So for this bulk flow over a long distance, the water potential gradient is due mainly to a gradient of pressure potential. Transpiration results in pressure potential at the leaf end of the xylem being lower than at the root end. So the values shown are just a snapshot. So it always goes from pressure. The movement of xylem sap against gravity is maintained by a transpiration cohesion mechanism. Okay, these are stomata, guard cells. The openings are called stomata. Here they're open, here they're closed. So these characteristics increase photosynthesis and increase water loss through stomata. So leaves have a very broad surface areas and high surface to volume ratios. So in the tropics, the leaves have a tendency to be bigger because there's more photosynthesis and more stomata, more leaf area. So plants lose a large amount of water by transpiration. So if this water that is lost is not replaced, the water will wilt. So transpiration also results in evaporative cooling when it's really hot. So it can lower the temperature of the leaf and prevent denaturation of various enzymes involved in photosynthesis. So when it's really hot, the stomata also close. And that's why C4 and CAM plants evolve for hot, arid climates. So about 90% of the water a plant loses escapes through stomata. Okay, each stoma has these guard cells, which control the diameter of the stomata by changing shape. So here the cell is flaccid, it's closed, here it's turgid, taking up water. You can see the vacuoles are bigger here than here, and they are open. Okay, and this is regulated by the cellulose microfibril. So changes in turgor pressure that open and close these stomata result primarily from reversal uptake. Okay, so here water going in, potassium ions, and loss of potassium ions. So potassium plays in here, the red dots. Okay, so here they are going in, going out, potassium ions going out. Okay, there are different types of plants that have evolved for arid climates. They are called xerophytes, okay? And they have these areas here. Okay, this is an oleander that lives in Florida, hot tropical climates. It has a thick cuticle and multi-layered epidermal tissue here to protect it, okay, that reduce water loss. The stomata are in these cavities here called crypts. So you can see them here. Again, this still is to prevent against water loss. So it protects the stomata from hot, dry wind, helps minimize transpiration by blocking the flow of air. Okay, so this has a higher humidity, this crypt, than the surrounding area. Okay, these depressions. So these are modifications. Stomata are concentrated on the lower leaf surface and so translocation is the transport of organic nutrients in a plant. So organic nutrients traded through the phloem. Okay, sugar source, where photosynthesis is, to sugar sinks, where the sugar is needed for growth. So phloem sap is an aqueous solution that is mostly sucrose. It travels from where it's made to where it's utilized, sugar source to sugar sink. A sugar source is an organ that is a net producer of sugar, such as mature leaves where there's a lot of photosynthesis. And a sugar sink is an organ that is a net consumer for growth, or it can store the sugar, like in a bulb or a tube. So active transport in phloem requires active transport, just like we talked about the proton pump, the co-transport of once hydrogen ions are pumped out of the cell, then co-transport back with the sucrose going with the hydrogen ions flowing down their concentration gradient. And this enables to accumulate sucrose. So in studying angiosperms, flowering plants, researchers have concluded that sap moves through a sieve tube, okay, by bulk flow driven by positive pressure. So here's the transpirational pull up the xylem. Here's the pressure down here from greater to lower. Okay, so sucrose in, water in, hydrogen ions in, and this out. Okay, so the source to the sink. Okay, so loading of sugar into the sieve tubes reduces the water potential inside here, okay, in the sieve tubes. And this causes the tube to take up water by osmosis from the xylem. Okay, so water flows in. The uptake of water generates a positive pressure that forces the sap to flow down the tube. And the pressure is relieved by unloading the sugar and the consequent loss of water at the sink, okay, which is down here. So here it's being stored. The sucrose is being stored in the sink. And then in leaf-to-root translocation, xylem recycles water from sink to source. So it goes from here, and back over here. So pressure flow in angiosperms. Okay, so the pressure flow hypothesis explains why phloem sap always flows from source to sink. And there have been experiments have built a strong case for pressure flow as the mechanism of translocation in angiosperms. Okay, so here a sap droplet in an aphid feeding, a stylet in a sieve member tube here, and a sap droplet, severed stylet from an insect, and it's exuding sap. Okay, so an aphid probes with this hypodermic-like mouth part called a stylet here, and it penetrates the sieve tube, which you can see right here. And the pressure forced out of phloem into the stylet, then when you separate this aphid from the stylet here, it exudes sap for hours. Okay, so the sugar concentration of sap from stylets at different points between a and a sink.